All right. I'll zoom in and it'll get better. All right. So this is a chloroplast. All right. This whole structure is your chloroplast. All right. If you notice over here on the side, it's got a double phospholipid bilayer. So this is actually not a cell wall. It's just a two phospholipid bilayers. Now, what is and so what do we need to get photosynthesis started? We need light, right? We've got to have the light turn on. Okay? So that's actually what these little zigzags represent. They're representing light. So I'm going to take and I'm going to write over here. Oh, come on. That's good. All right, I'm going to write here light energy. Bless you. And I'll write one on this too. And I'm writing light energy because it's not just sunlight. I mean, I could grow plants in here, although I'd probably kill them because I'm not a green thumb. But as long as PG&E doesn't shut off my electricity, right?
That's not something reliable.
Yeah.
It's not reliable, I know.
Yeah.
The sun is a little more reliable for at least another 4 billion years or so, right? All right. So we've got light energy. So it's going to, the photons are going to come through and into my chloroplast, right? Now they're going to end up at our photosystems, and we'll talk about that in a moment. What's the other thing that we need to have to do photosynthesis? We need water. Now, how many waters do we need to do photosynthesis? Just to make one glucose molecule. We need 12. All right? So up here on this little line that's coming in, I'm going to write 12 H2O. This is going to represent my 12 waters diffusing out of the cytoplasm into the chloroplast, and then they're going to come down here into what structure? Do you remember what this was?
Thylakoid.
It's a thylakoid. All right. So now I've got a bunch of thylakoids stacked up here. So I'm just going to write a little arrow. I'm going to draw an arrow here and here. What am I doing? All right, and write thylakoids. So I remember that that's what I'm looking at. And when I stack my thylakoids, what do we call that stack? Do you remember? Grana, that's right. It's called, a stack of thylakoids is called a grana. So I'm going to do one of these little doohickeys. Watch.
Dude!
All right. And I'm just going to put grana so that I know that that big stack is a grana. I've got a grana, stack of thylakoids, grana. Okay. All right. Yes. You
Thomas.
made it! Oh, Thomas!
Hi, Thomas.
You
We're all
go.
waiting for Thomas.
You got that, Jack?
What that?
And why can't I get it? Thomas is here.
Yeah, you're right.
Yeah, I
Yay! Okay. Now I'm not going to worry about Thomas. We're just going to worry about both. All right.
I changed that.
Okay, I'll worry about it.
Yay. I'm special.
Yes, that's what you want. All right. So I've got my 12 waters coming in, okay? And my light energy, all right? Now my light energy, I'm going to interact with my photosystem 2 here first, okay? You can't see. Maybe if I move it like this, you can see better. All right.
It's the same thing.
You still can't see? I'm sorry. All right, so.
You are blinded.
Aiden, you forgot
All right.
to press record.
So my light energy is coming in here to photosystem 2. So I'm going to write a capital P and a Roman numeral 2 to represent photosystem 2. So I remember that. All right. Now, at photosystem 2, when that light energy strikes it, it's going to interact with the chlorophyll that are in there. Now, there are thousands of chlorophyll molecules in each photosystem, right? And when the photons are going in there, they're hitting them and exciting the porphyrin ring and that magnesium. Those magnesiums are losing their electrons, right? And so what you don't see here is all along outside of this, there are these enzymes, all right? And the enzymes actually respond to the change in polarity that occurs. When you remove the electrons here and they pop out, they're going to go down the electron transport chain, and we'll talk about those in a minute, all right? When they leave, you change the polarity of this enzyme, and then that causes a reaction by the water-breaking enzyme. Right, that water-breaking enzyme is going to grab a water and break it, right? Rip it to shreds, right? So we're going to take, here I've only written one water, right? But this is really happening 12 times. So we're going to take our water, we're going to break it, and we're going to get 2 hydrogens, all right? Now those hydrogens also lose their electrons when we break it. So they're actually protons, H+. All right? Now, those 2 electrons that came off, they're going to go replace those electrons lost by the magnesium here in photosystem 2. So I've got 2 electrons in photosystem 2. For each water molecule, I'm getting 2 electrons replaced. All right? Now, so I've got my hydrogen, their electrons, so this is gone, the H2 is taken care of. Now I've got this O left, this oxygen. Okay, now, how many oxygen atoms will I have? No, total. I'm going to have 12, right? Okay, so what's going to happen here is when I break this one water, 1 oxygen atom's going to hang out. Then I'm going to break the next water molecule, and I'll get 2. So they're going to come together, and they're going to make O2, which is oxygen gas. It's relatively stable, right? So the O2 is going to diffuse out of the thylakoid, through the stroma, out of the chloroplast, and into the plant cell. Now I started with 12 waters. So how many oxygen atoms do I have?
6.
No, how many? Well, okay. How many oxygen atoms do I have? 12. All right. So I'm going to make how many molecules? 6. All right. So this becomes 6O2. Now, in the equation that you've learned in the past, how many oxygens do you get when you make glucose?
6.
6, right? You always learned 6, right? But how many waters in that old equation did you put in?
12.
No, you put in 6. You were always taught that it was 6 waters plus how many? What is it? 6 carbon dioxides get you 6 oxygens and 1 glucose, right? That's all nice and balanced and everything. But guess what? All of the oxygen that you get out of this, so all of the oxygen that you get to breathe, right, comes from this transaction here. So if I only started with 6 waters coming in here, how many oxygens would I get? 3. I'd get half as many. So that equation can't work. So this is why you're relearning in my class that it's really 12 waters going in. Because you cannot get this result, this 6O2 result, without starting with 12. Okay. So, we've made our oxygen, so now we can breathe, right? But now we've got to deal with this, what we're creating inside here. Now, that's better. All right, So we've got these electrons that are coming in here, right? And we're breaking off, every time we break a water, we're getting 2 protons in here. These electrons now, they're gonna get excited by the photons, and they're gonna have to move out. And then we're gonna get new ones coming in. And then they're gonna get excited and move out. We're gonna get new ones coming in, all right? So it just keeps going. As long as we have light coming in, the electron movement continues. Now, when the electrons move, they're going to go down the electron transport chain, right? So they're going to move down the electron transport chain to photosystem 1. Now, as they are moving down this electron transport chain, right, for each electron that moves through it, this central protein here is actually a proton pump, right? So it's going to move protons from outside in the stroma to the inside of the thylakoid. So each time an electron runs through here, we're going to get a proton being pulled in. So essentially how this proton pump works is when that electron gets in there, there are protons hanging outside here in the stroma, and we'll talk about how they get there in a minute. Remember, they're protons. They're a hydrogen that's missing their electron. They want it back, right? And as that electron goes through there, that proton pump kind of teases it, all right? It's like, here's an electron, and the proton goes, hey, and goes through as the electron passes the other way, and then it's like, now the proton's stuck in here and the electron's long gone. Alright, too bad, psych. Alright? So, every time an electron comes through, you get a proton pulling in. So I'm pulling protons in. So I get a proton, electron, proton, proton, proton. Right? So I'm building up a concentration gradient of protons every time. Now for for the 12 water molecules that's going on, I'm getting 24 just for breaking it, and I'm getting 24 from the electrons moving. So I'm getting, for every 12 water molecules, or for every glucose I make, I'm getting 48 of these protons building up inside. So I'm building up quite a concentration gradient. Okay? So I've got all these protons inside, right? And now what do they want to do?
Leave.
They want to leave. They want to become
Equal.
They want to become equal between, they want the inside of the thylakoid to be the same number as the outside. And they only have one escape route, and it's through this guy here. All right? This enzyme. It's called, you guys remember? ATP synthase. So I'm going to do ATP and then synthase. Try to make it fit in here. Hopefully you can read my chicken scratch. All right, I've got ATP synthase. All right, so my protons that are stuck inside this crowded space, all right, they're going to move through the ATP synthase and out into the stroma. Okay? Now, remember, as those protons go through that ATP synthase, right, it's actually like a corkscrew shape inside. So the force of the momentum as they go through is going to cause this ATP synthase to begin to spin. Right? And as it spins, it's got these little rotors on it that are going to pick up an ATP, or adenosine diphosphate, so it's got 2 phosphates, plus a phosphate group. Right? So it's going to pick up an ADP, then it's going to pick up a phosphate group, it's going to slam them together, and we're going to go from having 2 phosphates to 3, so it becomes adenosine triphosphate, or ATP. All right? Now look, the way I drew this phosphate group here, I did it with a P with a circle around it. but in some of the literature you have that I've given you, you're going to see a P with a lowercase subscript I. Right? It's the same thing. The P with the circle, the P with the little tiny I, same thing, but what I found when I did the drawings with the I, it became a hot mess. So it gets smudged and it's hard to tell what is that. So the circle's a lot easier to figure out what's going on. Yes?
One ATP is made for every proton?
So it in a perfect world, yes, one ATP should be made. But what ends up happening is it's slightly less. Because what happens is as this is spinning around, from every time one goes through, it's making a rotation, but there may not be an ADP or a phosphate, so it'll skip a turn until it picks it up. So when we do all the calculations in the lab, it ends up not being quite, but it's close. So yeah, and that's what we suspect is really happening there. Is it's spinning, and when the rotors go to grab it, it misses it or it's not there, and so it just spins around the next time, grabs it, and then puts it together. So on average, it's slightly less than 1 per hydrogen. Yeah. All right? But that's microbiome. You guys don't need to know that. All right. So, but it's essentially that. Okay, so we've got these hydrogens now that have escaped, right? We've got these protons that come out. Now these guys are out in the stroma and they're floating around. Now, they've got some choices. They got 2 choices, right? They're going to float around, and then they're going to get stuck being this guy, getting sucked back in. All right. So just finally escaped, and now it's trapped back in again. Darn it! All right. We got to do it all over again. Or he's going to end up over here. We'll talk about that. All right. Remember we had those electrons that were coming down here to photosystem 1. Now essentially they've lost their momentum at this point. It's a very complex physics thing. Don't worry about it. Once they get here, they're going to get essentially re-energized. They're going to get kicked back out again and go down a mini electron transport chain that's going to take them to this enzyme here. Right? And this one is called NADP plus reductase. So those electrons are going to move down to NADP plus reductase. REDUCTASE, reductase. All right. Now, NADP plus reductase is going to take 3 things and smash them together. Right? So it's going to take NADP plus, which is just our hydrogen taxicab that's inside the chloroplast. Now, I've had this question all day. So the NADP plus is made by the chloroplast, but they only make a certain amount. It's a recycled molecule, so they don't need to keep making it. Right? They make probably, 15,000 of them or whatever, and then they're just little cabs that are going to go back and forth to the Calvin cycle carrying hydrogen all day. Right? They're going to drop off the hydrogen, come back. Drop off the hydrogen, come back. So this is a recycled molecule, just like ADP. ADP is a recycled molecule. It's going to make ATP, the ATP is going to get used, and then we got ADP left over, and then we're going to make more ATP. Okay? So the NADP plus, we're going to add to it a proton. Right? Now, if you look here, I've got NADP plus, I'm missing an electron, and I've got a proton that's missing an electron. So these 2 electrons here are going to move through here, then they're going to get pushed over here, and then they're going to fill these 2 spots. So that gives me NADPH. So I've essentially taken my NADP plus molecule, slapped an electron on it, slapped a proton on it, and put an electron into that. All right, so I got NADPH. Now my NADPH and my ATP are going to be used in the Calvin cycle, right, to help make my triose phosphate, which will end up being glucose. All right, so let's look at this again. We've got our light coming in, right? We've got our water. At photosystem 2, we're going to break apart our water. We're going to take the electrons into the photosystem to replace those lost in the magnesium. We've got protons being released into the interior of the thylakoid. And we've got oxygen that's going to meet up with another oxygen and diffuse out as a gas. Now that oxygen, when it gets out here into the plant cell, it can be used by the plant to do cellular respiration, but there's so much of it being produced that the plant doesn't use it all. And so we get the benefit of that, right? The excess oxygen just diffuses out of the plant and into the atmosphere, and right. So these electrons, they're going to move down the electron transport chain. We're going to bring more protons inside, so we get a nice concentration gradient going in here, right? And then we're going to use the diffusion, the facilitated diffusion of that hydrogen out through ATP synthase to spin it and make ATP. These protons are either going to get sucked back in or get put, eventually they're going to get put onto the NADP plus to make NADPH, right? Along with the electrons that were coming down the transport chain. So we've made NADPH and ATP for use in our Calvin cycle. Aiden.
So they just make ATP just by the thing spinning.
Yeah, because what I didn't draw on here, it's got these cute little things, they're like stator rotors, or they're little stators. And they're gonna, as it spins around, they're gonna hook on to an ADP and hook on to a phosphate and then push them together. Just by spinning. Yeah. So the energy that we're using to make the ATP is really just the momentum of the hydrogen as it's escaping, as it's going from high to low. Now if I turn out the lights, my electrons stop moving because they're not getting kicked out anymore, and the whole system stops. This runs down, and then there's no more concentration gradients and no more ATP, and eventually my NADP plus gets filled by the hydrogens, and then we just wait until the light comes back on. All right? All right. Any other questions? All right, I'm expecting questions for sure on Monday, I assume. So what you want to do is walk through this. You're gonna talk to yourself, and your parents are gonna think you're nuts. All right, let them. All right.
You're gonna do my
H2. You want to know where that hydrogen's going. Follow the hydrogen. Talk to yourself. Where does it end up? What is it doing? Where is it going? How is it flowing through this? Look at the oxygen. Where does the oxygen go? All right? Where does it end up? All right? And then you're going to take your notes. You're going to look at the slides. And you're going to look at the light-dependent reactions. That's what this is. All right? So let's make sure we write that down. Light dependent reactions or phase. All right, same thing. All right. Look at that in your lecture notes, and as you read through what I've taught you, look at this. Follow it. Follow the molecules. Right? This is what's going to be key to you doing well on the test. This is what I want to know. Do you know where it's going and why? Okay. Why is water interacting with photosystem 2? Big whoop. All right? That kind of thing. Okay? Any other questions? Anything? Anyone? Bueller? All right, you guys have the rest of the period. It's Friday. Enjoy.
Oh, Friday.
You just can't wait, kid.
I come to play.
Cellular's like the best. Cellular's like the best movie ever.
Maybe you should post that for